Mobile elements are one of the major sources of human genetic instability. Understanding their expression in different tissues and conditions is critical to understanding their impact on the genome. The vast majority of L1 transcripts are the result of passive inclusion of L1-related sequences in other transcripts that have no role in the L1 life cycle. Our approach eliminates this irrelevant background.
This protocol can be adapted to studies of any mobile element, or even viruses in any sequence genome. There needs to be at least some sequence variation to allow discrimination between loci.
Visual demonstration of this method is critical in illustrating the stringency and care required to confidently identify expressed L1 repetitive elements at the locus-specific level.
Begin this procedure with cytoplasmic RNA extraction and next-generation sequencing as described in the text protocol. By selecting for cytoplasmic RNA, L1-related reads found within expressed intronic mRNA in the nucleus are significantly depleted. In the sequencing library preparation, another step taken to reduce transcriptional noise unrelated to L1s includes the selection of polyadenylated transcripts. This removes L1-related transcript noise found in non-mRNA species. Run alignment paired-end sequencing FASTQ files with the RNA-seq sample of interest using bowtie1 by typing the command line in the Linux terminal. This alignment strategy requires that transcripts be uniquely and collinearly aligned with an exhaustive genomic search. The strategy provides confidence in the calling of reads mapping specifically to a single L1 locus. Strand separate the output BAM files using SAM tools and Linux commands to select for the top strand and the bottom strand. Note that the